Hello, welcome to the Connect Science series. We have someone very special. I'm very excited to host today Dr. Cedric Blampang. I hope I'm pronouncing it right. My French is not really good. And I'm gonna give, so before I go into the introduction, please type your questions in the Q&A option, not in the chat. And the seminar will get recorded and is going to be posted in the website from the Connect series, where you can see each week's presentation. And now the introduction. So Dr. Blampang is the director of the Lab of Stem Cells and Cancer and a distinguished professor at the Free University of Brussels, where he actually got his MD and PhD. He's actually, he did his residency in internal medicine, which I didn't know. I found it very interesting. So after his initial training, he moved to New York to the Rockefeller University, where he did his postdoctoral training with Dr. Elaine Fuchs. He was amongst the people that discovered hair follicle stem cells and stem cells in the skin, like pioneering that work. And after that, he moved to Brussels in 2006, starting his own lab. And after that, he has done amazing, truly groundbreaking work, not only in the field of stem cells, but also in cancer and discovering the cell of origin in many tumors, not only in basal carcinoma or squamous cell carcinoma, the skin tumors, but also in breast cancer. So he jumped into mammary gland stem cells, which is amazing. He has done lots of work in cancer resistance, stem cells, and more recently, he has also very, very interesting and very elegant work with lots of elegant mouse models in EMT, how EMT is regulated, like the various transitions. So lots of amazing work, and I could talk for an hour about his work because I like it so much. I'm not gonna do it, I'm gonna let him do it, who is the one that should do it. So thank you a lot, Dr. Blampang. I'm so thankful that you accepted our invitation. Thank you so much.
Thank you very much, Judith, for the invitation, and thank you very much for the very kind introduction. I'm very honored to be here today. And as Judith said, I'm going to discuss with you the mechanism that regulate different tumor transitional states. So as you may know, within the tumor, not all cells are alike. Some cells proliferate, some cells are differentiated, some cells invade the surrounding tissue and can reach the circulation and leads to metastasis. And upon treatment, some cells may enter into a quiescent stage and resist to the therapy. And so these differences between the different tumor states are called inter-tumor heterogeneity. And many different mechanisms have been proposed to regulate this inter-tumor heterogeneity, which has a very important implication for the prognosis and the therapy of the patient with cancer. And so I will illustrate this very important question in a mouse model of squamous cell carcinoma. So the squamous cell carcinoma is the second most frequent skin cancer, and it affects about 1 million of patients per year throughout the world. In most of the cases, it can cure by surgical excision, but nevertheless, in a fraction of the case, it can lead to metastasis and then, associated with much poorer prognosis. But more importantly, squamous cell carcinoma is also the major tumor type in oral cancer, in head and neck cancer, in esophagus cancer, and in a fraction of non-small cell lung carcinoma. And in this location, the prognosis is much more poorer, and it can lead to many deaths of the patient each year. So in the skin squamous cell carcinoma, the two major oncogene and tumor suppressor gene are mutation in the RAS pathway, and can be HRAS, it can be KRAS, it can be RAS2, and it's also accompanied by mutation in the p53 tumor suppressor gene. And the first question that we're trying to address in our laboratory concerning tumor heterogeneity of the skin squamous cell carcinoma is the tumor heterogeneity can arise from the cell type in which the cancer begins. So in other words, is the cancer of origin can somehow prime the tumor to adopt different tumor phenotype. And to address this question, we use a lineage tracing strategy in which we can activate KRAS G12D and p53 deletion, as well as the labeling of this cell with YFP in different cells of the skin epidermis, and check whether this different cell of origin can lead to different tumor type. And to our great surprise, we found that the activation of KRAS and p53 deletion in the interfollicular epidermis, the part of the skin that is responsible for the skin barrier, leads to the development of what we call a well-differentiated squamous cell carcinoma. And so well-differentiated squamous cell carcinoma is very easy to identify by any pathologist in the world. When they see these keratin pearls, they would see that, you know, this is a squamous differentiation, and therefore the type of cancer is this well-differentiated squamous cell carcinoma. And when you stain these YFP tumor cells with epithelial markers such as keratin 14, you see that all the cells here are positive for K14. And when you stain for a mesenchymal marker that marks the stromal cells, none of the tumor cells here express vimentin, which is only expressed by the cancer-associated fibroblasts and the other stromal cells in these tumor groups. So, in sharp contrast, when we activate KRAS mutation and p53 deletion in hair follicle stem cell, we found that most of the tumor that arise from this hair follicle stem cell were what we call a mixed SCC. So a mixed SCC is a part of the SCC is composed of epithelial tumor cells, here as stained by keratin 14, but another part of the tumor had lost the expression of K14 and gained the expression of vimentin, which is a null mark of epithelial to mesenchymal transition. And when we analyze the expression of EpCAM, which is, it's an epithelial marker in all well-differentiated SCC, you see that there's a monomodal expression of high level of EpCAM expression, in good accordance with their epithelial phenotype. But when you look at the mixed SCC, you see that there was a bimodal expression of EpCAM. EpCAM was expressed in the epithelial part of the tumor, but was lost in the mesenchymal EMT part of the tumor. And you see here, when there's several hundred of tumors that are plotted in this violin plot, you see that almost all tumors are present to some degree EMT. You can have a very low proportion of EMT, like here, like 50% of EMT, or a very high proportion of EMT, as you can see here, which will lead, when everything is EMT, it's what is called a spindle cell squamous spindle cell carcinoma. So what I show you here is that actually the cell of origin matters, and in driving tumor heterogeneity, and the well-differentiated part of the skin, the interfollicular epidermis, lead to a well-differentiated SCC, whereas the hair follicle stem cell and its progeny can lead to these EMT tumor cells composed by an epithelial part and a mesenchymal part of the tumor. These EMT tumor cells, or the occurrence of EMT, has a very important consequence for the progression of this tumor. And when we check the proportion of mice with lung or lymph node metastasis in this hair follicle-derived tumor, we found that about 30% of the mice present a lung metastasis, and about 50% of the mice present lymph node metastasis. Where, in contrast, the interfollicular derived SCC with low rate of EMT, they present a very low incidence of metastasis, either lungs or lymph node. And in all cases that present metastasis, some of the tumor did present a certain fraction of EMT. So in this skin model of SCC, metastasis is invariably associated with EMT. I don't mean by that in all cases where you will find metastasis, EMT is always the driving force of that. But in this particular model, it seems to be the case. Something that people have, noticed already for a while, is and which kind of make the role of EMT during metastasis sometimes controversial, was the fact that when you look at metastasis, most of the metastases do not present any sign of epithelial to mesenchymal transition. And this model doesn't make an exception to this phenomena. And when we check the lung metastasis here, we found that 70% of these lung metastases did not present any sign of EMT, and no cells express vimentin. In a small proportion of the case, there is an expression of K14 and vimentin, and a very, very low frequency of there's only cells that did not present sign of epithelial differentiation. So suggesting that mesenchymal to epithelial transition is important for lung colonization and metastatic outgrowth at the distant metastatic site. Another very important functional consequence of epithelial to mesenchymal transition is the resistance to therapy. And in our laboratory, we found that indeed, if you administrated cisplatin or 5-FU to mice that present these EMT tumors, we found that the EMT, the epithelial part of the tumor were really sensitive to chemotherapy administration, as seen here by the proportion of active caspase-3 24 hours following the administration of the chemotherapy. Whereas the mesenchymal part of this tumor, the EpCAM negative tumor cells, they present very low proportion of active caspase-3. And this was also observed in vitro and to not only to cisplatin 5-FU, but to a broad range of chemotherapy or molecule that induce replicative stress. And so I'm not going to discuss that, but we have dissected now the underlying mechanism behind this increased resistance of therapy of the EMT tumor cells, and we hope to be able to present you that in a next seminar in the next slide. So another burning question when we look at EMT in these tumors, as in many different tumor settings, does EMT occur through a binary process in which like this EpCAM expression would suggest, or do EMT occur through distinct transitional state? And to address this question, we use cell surface marker screening to use a bunch of different monoclonal antibody and check whether the EpCAM positive tumor cells or the EpCAM negative tumor cells, do these tumor cells express homogeneously this different cell surface marker, or in contrast, do some of these tumor cells present an heterogeneous expression of this different marker that would suggest that it's not a binary process, but maybe through distinct tumor state. And when we did that in our skin SCC EMT tumor model, we found that the epithelial part of the tumor were very homogeneous, and most of them express an homogeneously expressed the different cell surface marker that we screened. But the EMT tumor cells, in contrast, were very heterogeneous, and most of the tumor cells, they do express heterogeneous expression of this cell surface marker. And as you can see here, there's a bimodal expression of these different cell surface marker. Then we use this cell surface marker first 2 by 2 and then 3 by 3, and to try to deconvolute, if you wish, the heterogeneity that were hidden in these EpCAM negative tumor cells. And using 3 cell surface marker, we found that these 3 cell surface marker reliably can identify 6 different populations of EMT tumor cells. And so, and which are these 3 cell surface markers? CD51 and CD61 are alpha V beta 3 integrin. And so those molecules were previously found by other laboratories to mark cancer stem cells or cells at the invasion front in certain type of tumor, including breast cancer. And CD106 is VCAM1, which was found by the group of Johan Massaguet as a marking a subpopulation of metastatic initiating cells in certain type of cancer. So now, having these different cell surface marker, we could isolate these 6 different populations and stain them with either K14 or vimentin to assess their degree of differentiation. And as you can see here, as soon as you lose EpCAM expression, tumor cells begin to express vimentin. But at the beginning of the EMT process, most of the cells co-express actually epithelial and mesenchymal marker at the same time. And we call this tumor hybrid tumor cells, hybrid EMT tumor cells. Other people call that partial EMT, quasi-mesenchymal population, or this term refers to this cell population that co-express at the transcriptional level and at the protein level epithelial and mesenchymal program. And as you see here, along the EMT progression, cells progressively lose the expression of the K14 and only express vimentin expression, as you can see here in this triple positive CD51, CD61, CD106 population. So it has been previously suggested and shown that EMT may be important for metastasis, but whether the late EMT state or the early EMT state were the most important for metastasis was not very well understood. So we had here an ideal model to test this hypothesis, and we inject in tail vein this different fraction of the different EMT tumor population. And what we found here is not at all the most EMT population that was the most metastatic cells. It was this early hybrid EMT population, such as a triple negative or CD106 alone, that were the most metastatic population in this skin SCC. And when you check where in the tumor these different populations are located, actually we found that these different EMT states could be found in different areas of the tumor. And so as you see here in 3 different tumors, there were different type of tumor cells. In the area 1, as you can see here, the cells were really epithelial, very nicely cohesive, and expressing only K14 and not expressing vimentin, whereas in the area 2, you can see here the cells begin to be a little bit more elongated, but still connected to each other through desmosome, and begin to express in some part of the tumors the co-expression of K14 and vimentin, as you can see here. And more you go into this late EMT stage, the cells getting more and more elongated and eventually lose the expression of the K14 and only express vimentin or CD61. And when you check, the stromal composition of this different tumor, you found very different stromal composition according to the distinct EMT state. And so the very late EMT state present a very abundant infiltration of macrophages and other inflammatory cells. They present an increased number of blood vessels and in lymphatic vessels, as you can see here. So now I show you, to deconvolute, this different tumor state through the use of monoclonal antibody, but you can do exactly the same thing with single cell transcriptomics. So when you take such as skin SCC, you have the tumor cells that are here, and when you do a clustering of these different tumor cells, you can see that they can be deconvoluted in epithelial, early, hybrid EMT, more late EMT, and the very last step of EMT population. And using single cell ataxic, you find also the same kind of clustering with late, early, hybrid, and purely epithelial EMT, and you can, using the single cell ataxic, also identify, which are the enhancers that are specific for this different state, and what could be the transcription factor that are associated with the open chromatin region that you find in this different state, and also that is, we are working actively on that, and I hope to be able to update you the next time. So what I show you here is that the EMT the EMT process is not a binary process and is accompanied by different states. Some of them are more stable than the others. So the early hybrid EMT state and the late mesenchymal tumor state are a little bit more stable than the other, whereas the state in between are very plastic and they can switch very easily to each other's. These different tumor states are, and I didn't show you that, express different set of transcription factor and are associated with different activation of different enhancer, and they present a very different tumor function. The most proliferative cells are the tumor epithelial cells, whereas the early hybrid EMT state are the most metastatic tumor population, and whereas for example, the late EMT tumor state are the most invasive one. And also these different states reside in different location, in different niches, and these different niches certainly contributed to the regulation of their transcriptional landscape. But whether so all these would be, a type of epigenetic regulation of EMT, and by the intrinsic property of the tumor cells, but also through their interaction with the microenvironment. But would it be possible that certain mutation can promote the acquisition of a very particular EMT state? That was, an open question. And we had sequenced some time ago the chemical induced skin SCC, very frequently, probably the most ancient tumor model, in which you treat the mice with a carcinogen, DMBA, and then you follow by the application of TPA, and in this tumor protocol, first benign tumor occur, and a fraction of this benign tumor will transform into an invasive squamous cell carcinoma. But in this invasive squamous cell carcinoma, the vast majority of them consists of this what we call well-differentiated squamous cell carcinoma. And when we sequenced them, we found, and we were pleased to find that actually we found the same mutation that were also found naturally in human SCC from different origin, from the skin SCC, head and neck, lung SCC, esophagus SCC. And our attention was caught by different of this molecule that were mutated there, and for which the mechanism by which they regulate tumoralization were not known or very poorly known. And so we decided to move ahead, and we are currently studying a certain number of these genes. And among these genes, this part of my talk will be focused on this protein, which is called FAT1. And FAT1 is a protogadarin, and it's heavily mutated in many different human SCC. Is the second most frequently mutated gene in head and neck SCC. Is among the most frequently mutated gene in the lung SCC, as you can see here. In oral SCC, it's also the most frequently mutated, second most frequently mutated gene, with 40% of the case present mutation in FAT1. And when you look at where the mutation is, all these mutation, they are stop gain mutation, so truncating mutation, that most of them arise before the transmembrane region of this transmembrane protein, so leading to a non-functional protein that's not expressed at the cell surface. And so we decided to study FAT1 by performing the conditional knockout of FAT1 in the skin epidermis, and the mice with FAT1 mutation, they're born alive at, Mendelian ratio, and they present no phenotype. When we treat them with DMBA TPA, they present early occurrence of skin tumor formation, as you can see here, and they also present an increased number of tumor cells, tumors per mice. So consistent with the notion that FAT1 is indeed, a tumor suppressor gene. That's, not big surprise. When we acutely knock out FAT1 in pre-existing papillomas, so in this benign tumor, using a K14 CRER, which upon tamoxifen administration can lead to the deletion of FAT1, we found that, macroscopically, very rapidly, we see the transformation of this benign tumor into a malignant carcinoma. And when you check that by microscopy, the thing are really striking. So these benign papillomas are, it's a very well-organized tumor with a basal lamina, as you can see here, with laminin 5, a proliferative layer of basal cells here, like the normal skin epidermis, and a different layer of suprabasal terminally differentiated cells expressing keratin 10. And upon FAT1 deletion, within 2 weeks, as you can see here, you begin to see a change in the shape of the cells, and an effraction of the basal lamina, and a progressive decrease of differentiation. And within 4 weeks, most of the basal lamina was gone. All the basal cells now present this, loss of cell polarity and invasive behavior, and they do not anymore differentiate into a keratin 10 population, which is, an hallmark of this benign tumor. So really, very rapidly, FAT1 deletion in benign tumor transformed this benign tumor into a malignant squamous cell carcinoma. And that was, the most surprising and exciting part of this study. As I told you, in this DMBA TPA-induced skin SCC, most of the SCC consists of this well-differentiated pathology with the expression of K14, but do not express vimentin. But in FAT1 tumor, almost all the tumor cells co-express K14 and vimentin and present this change in morphology, where you see it's much more roundish, and we found by EM that they do not have anymore all the adherent junction. They do not anymore express icadherin, at least in the strong DMT part of the tumor, but they still communicate and attach to each other through gap junction protein. And so when we quantify this, degree of EMT, you see in this controlled DMBA TPA, it's mostly a monomodal expression of webcam, whereas in FAT1 derived tumor, you see this bimodal expression of webcam, and you see here, almost all of the FAT1 derived tumor present EMT. But when you check the degree of EMT by, for example, or a set of monoclonal antibody, they were all stuck in this early stage of EMT, consisting of this triple negative of CD1 or 6, and almost no tumors present the occurrence of this very late purely mesenchymal tumor cells in this model. And when we isolate these EPCAM-positive or EPCAM-negative tumor cells, because they have a YFP reporter gene here to allow to distinguish them, most of the tumor cells present this hybrid EMT and the co-expression of K14 and vimentin in these tumors. And interestingly, this early hybrid states that we found in this FAT1 tumor were associated with an increase of lymph nodes, the proportion of the mice with lymph node metastasis, but also the number of lymph node or lung metastasis per mice. So we did the transcriptional profiling of those FAT1 derived tumor and compare them to the control DMBA TPA. And as you can see here, the FAT1 derived tumor present this upregulation of many mesenchymal markers such as cadherin-11, PDGFR-NB, FAP, vimentin, and many ECM protein and all, the most well-known transcriptional regulator of EMT. And when you check at the expression of the epithelial gene, at least a fraction of the epithelial gene were still highly expressed in this FAT1-derived tumor as compared to the LGR5, the genetic model that I showed you in the first part of my talk, and many of the epidermal differentiation markers are there, and they overexpress or express many regulators of epithelial differentiation, such as the grenier-like protein or SOX2, and you'll see in a moment why that's important. So when we did these, we did the FAT1 deletion in a human squamous cell carcinoma that are not mutated for FAT1. They began to present this mesenchymal morphology, and they co-express keratin and vimentin. They lose the expression of E-cadherin or Claudin-1, but they still express this epithelial regulator that are P63 or SOX2, but they also express at the same time ZEB1, a transcription factor that are important to promote EMT. When we check in our collection of human SEC PTX, which all of them has been sequenced, we found that the FAT1 mutated the PDX from head and neck or lung SCC presents some hybrid EMT score, whereas you rarely see these hybrid EMT cells in at least in the collection that we screen in FAT1 wild type PDX. So to try to dig a little bit further into the mechanism that regulates this hybrid EMT state induced by FAT1 deletion, we perform ATAC-seq, which allow to map all the open chromatin region of the genome. And when we compare that to the control EPCAM-positive tumor cells from in wild FAT1 wild type mice, you see here that when you do a motif discovery in the enhancer that get upregulated, for example, in the region of the SNAIL1 or ZEB1, we found that there was a great overrepresentation of TET motif. And TET motif are transcription factor that relay the YAP-pentase signaling to the nucleus. And when we stain for YAP1 in FAT1 derived tumor, we found that there were, very strong upregulation of FAT1 toward the tumors as compared to the YAP1 expression in the control SCC. And during the revision of our paper, the different group has reported this connection between FAT1 mutation and YAP expression. And the group of Silvio Gutkin found that YAP1 in head and neck SCC, more the tumor present invasive characteristic, stronger YAP1 is expressed there, and he could show that when he expressed a dominant negative version of FAT1, he could rescue the tumor growth in this assay. And there was also a report that FAT1 mutated breast cancer presents a resistance to the CDK4 and 6 inhibitor through the activation of the EPO pathway. So we began to perform, double and triple knockout of FAT1, YAP1, and TAZ, and found that when you mutated YAP and TAZ in FAT1 mutated tumor, we decreased the late stage of EMT. And when we transcriptionally profile those triple knockout cell, FAT1, YAP1, and TAZ, we found that a lot of the genes that are well-known YAP-TAZ target gene were not upregulated anymore in this triple knockout, and we could uncover the EMT program controlled by YAP-TAZ that promote this hybrid EMT states. We, in the transcriptional profiling, and as well as in the ATAC-seq, we also found there was an overrepresentation of the SOX2 motif as compared to the LGR5, for example, EMT tumor cells. And when we stain for SOX2, you see normally SOX2 is expressed in the epithelial part of the tumor, but is lost in the EMT tumor cells. Whereas in the FAT1 knockout tumor cells, SOX2 it's still expressed even in this hybrid EMT states. And so we have previously shown together with the other group that SOX2 is marking cancer stem cells or tumor with increased clonogenicity upon transplantation in the well-differentiated SCC. And when you knock out SOX2 in this tumor cells, there was a decrease in a shrinkage of the tumors and a decrease of their clonogenicity as well. And so we mutated SOX2 in the FAT1 knockout tumor cells. And in what you see here, when you mutated SOX2, all the tumor cells now progress with this full EMT state, which they do not anymore co-express K14 and only express vimentin. And when we perform the transcriptional profile of the SOX2 and FAT1 mutation together, we found that the epithelial gene were no longer expressed in this double knockout of FAT1 and SOX2 tumor cells, consistent with the notion that SOX2 is controlling the epithelial transcriptional program that accompany FAT1 loss of function. So we were facing a very a priori complicated question, which is how the loss of membrane protein like FAT1 can induce the activation of YAP and TAZ, which lead to the expression of the EMT tumor states, and the activation of SOX2, which leads to this epithelial program. And to address this question, we use our human cell carcinoma cell line mutated by CRISPR-Cas9 for FAT1 and compare that to the isogenic cell line, and then check, what was the phosphorylation and protein expression that were changed between wild type and FAT1 derived tumors. And so when we check the phosphorylation that were differentially expressed between the different conditions, we can mine this phosphorylation site and check what are the known kinases that phosphorylate this precise site. And among the most upregulation of increased phosphorylation was the YES kinase, which is part of the SRC kinase, and it was super strongly differentially phosphorylated between FAT1 mutated tumor cells and wild type FAT1 cells. And the predict kinase that were phosphorylating YES was calmodulin kinase 2. So then we could use, so the first thing we indeed found that calmodulin kinase 2 was indeed increased, its phosphorylation was increased in FAT1 mutated cells. You can see here a total phosphosarc was strongly increased in FAT1 mutated tumor cells, and the YES was the predominant SRC kinase that was responsible for this increase in total phosphosarc. And when we treated with calmodulin kinase inhibitor, this FAT1 derived tumor, we could decrease the phosphorylation consistent with the notion that calmodulin kinase 2 act upstream of phosphosarc to promote SRC phosphorylation. And we then use SRC inhibitors to show that when we treat the FAT1 mutated cells with SRC inhibitor, no, we do not see anymore YAP in the nucleus, and we do not see anymore ZEB1 expression. And this dependency of YAP translocation and EMT transcriptional program by SRC kinase was also recently found by other group in a totally different context. So what I show you here is upon FAT1 deletion, calmodulin kinase get activated, which in turn phosphorylated SRC and kinase and YES in particular, which lead to the YAP pentase translocation to the nucleus and the activation of the EMT program. What I'm not going to show you is we also found that calmodulin kinase 2 phosphorylate EZH2, which normally deposits repressive mark around the SOX2 locus, and upon the and which lead to sustained SOX2 expression. And so when we inhibit calmodulin kinase, we also decrease this repressive mark, so we increase this repressive mark, and we get an upregulation of SOX2. So having understand somehow the cascade of kinase that leads to this hybrid EMT differentiation program in this tumor cell, we check whether we can exploit that to see whether these tumor cells present increased sensitivity to this different inhibitor. And we were very pleased to see that two SRC inhibitors were the FAT1 mutation in those human SCC cell line render these cells more sensitive to these two SRC inhibitors as compared to their isogenic FAT1 wild type tumor cells. And so we could even show that this sensitivity to SRC inhibitor was also could also be found in vivo, and upon transplantation of this human FAT1 or wild type or FAT1 mutated tumor cells, the administration of SRC inhibitor prevent the growth of this tumor, where there has little effect on the FAT1 wild type. So one can imagine that we can induce like a kind of a personalized therapy for the number of patients with FAT1 mutation in the different that present FAT1 mutation in the different tumor contexts, and treat them with SERC inhibitors. So the current SERC inhibitors are a bit, toxic, although, some of them I think are used to treat, or at least in clinical trial, to treat some leukemia. There's another generation of SERC inhibitor that seems to be much more specific and much less toxic than the previous one, and we're trying to collaborate with the people who have generated this new SERC inhibitor and check first whether this SERC inhibitor would prevent PDX presenting FAT1 mutation, and if that's the case, we can think that we can try potentially to administer that to the patient in clinical trials with patient with FAT1 mutation. So the last part of my talk is going to be very brief there. I need to disclose that I have potentially a conflict of interest in this part of my talk, since I'm the founders of ChromaCure, which is a spin-off of the university, for which this part of the talk is related to. And the question that we had here is, what are the mechanisms that regulate the malignant tumor state? And so how do you move from a benign papilloma to a malignant squamous cell carcinoma? And it's been a long time that we have started this experiment, and we profile CD34 cancer stem cell of the benign and the malignant squamous tumor, and check what are the difference between in the transcriptome of these benign and malignant tumor cells. And we found that among the rare transcription factors that were upregulated only in malignant tumor cells was a gene called NR2F2. And NR2F2 was a well-known nuclear receptors, and that is very important during embryonic development, but not anymore and so the deletion of NR2F2 in adult mice had absolutely no phenotype. And the Tsai group have found that the NR2F2 was expressed, in addition to be expressed in certain type of cancer, it's also expressed in the endothelial cells of the tumor. And they show that the deletion of NR2F2 in the endothelial part of their tumor were sufficient to prevent or decrease strong D tumor formation. They also show that in a prostate cancer model, the deletion of NR2F2 was blocking the appearance of cancer cells. So suggesting that indeed NR2F2 could likely regulate this malignant transition in this skin squamous cell carcinoma model, in which, in addition to be expressed in the endothelial cells, expressed in heterogeneous manner in the tumor cells. And we obtained the flux allele from the Tsai group and cross that with the K14-3, which was totally viable and present absolutely no phenotype. When we treat this conditional knockout on NR2F2 in the skin epidermis with the DMBA-TPA, we found that the benign tumor, their number was more or less similar between the control and the NR2F2 knockout cell. But these mouse did not progress into a squamous cell carcinoma. And the same was also true with the KRAS P53 genetic model that I show you here, in which when you induce the deletion of NR2F2 in this model, normally this model rapidly develop only invasive squamous cell carcinoma. They don't pass by this step of papillomas. But in the NR2F2 knockout, the number of tumors per mice were relatively similar, but it was exclusively composed of benign tumor cells and almost no invasive squamous cell carcinoma. So these two data really suggest that NR2F2 is an essential regulator of malignant transition. We generate a new mouse model here, allowing the overexpression of NR2F2 in this benign tumor or in benign papillomas, and very rapidly following NR2F2 expression, the tumor cells changed their shape, effract their basal lamina, and became to overexpress marker of malignant transition, consistent with the notion that indeed NR2F2 is very important for malignant transition and the regulation of malignant state. We performed the conditional deletion in existing invasive squamous cell carcinoma and transplant them to immunodeficient mice to assess their tumor propagating cell frequency. And we found that the deletion of NR2F2 drastically decreased the number of secondary tumor upon transplantation in this mouse model, suggesting that, and in 3 different human cancer cell lines, squamous cell carcinoma, skin, lung, and esophagus, the deletion of NR2F2 strongly decreased the ability of these tumors to reform secondary tumor upon transplantation. So that shows you that NR2F2 promotes tumor stemness or tumor propagation in both mouse and human SCC. Most importantly, for the therapeutical perspective, we found that when we acutely deleted NR2F2 in pre-existing invasive squamous cell carcinoma, these tumors were rapidly shrinking. And after 3 weeks, almost all the tumor had shrunk. And when we section, those rare tumors, they were composed of necrotic or completely terminally differentiated material, suggesting that NR2F2 is essential for the maintenance of the malignant tumor states. When we overexpress NR2F2 in these benign tumors, we found that the expression of the differentiation of the tumor cells, such as keratin 10 here or ikaderin here, were rapidly decreased following overexpression. And something that was really striking, and for which I really did not expect that, when we stained for keratin 10 in this invasive squamous cell carcinoma, following the deletion of NR2F2, there was a massive upregulation of keratin expression, keratin 10 expression, which normally are never found in invasive squamous cell carcinoma. So somehow you can retransform and promote the differentiation of a invasive malignant tumor into a benign, terminally differentiated tumor cells. And when you stain here, and that's an HNE, when you stain here with this tumor with an HNE, you see that you almost transform the entire tumor into this keratin pearls in keratin amorphous material. And so that suggests that targeting NR2F2 can promote the differentiation of the tumor cells into these non-tumorigenic cells. And so this differentiation therapy, although has been proved to be very effective in certain type of leukemia, it's still very rarely used to treat solid tumors. And so that's what ChromaCure is trying to develop. It's small molecule inhibitor that would inhibit NR2F2 function in cancer cells. So we then try to understand the molecular mechanism behind the function of NR2F2 in these squamous skin tumors, and we found that there was a bunch of genes, about 200 genes, that were both upregulated following gain of function or downregulated following loss of function, and those genes encompass regulator of cancer stem cell, ECM molecule that are involved in angiogenesis and in lymphoangiogenesis, and many molecules that are involved in tumor progression and EMT. And for example, when you check when you overexpress NR2F2, for example, here you can see that leads to the upregulation of ZEP1 or ZEP2 expression, you know, two very well-known transcription factor regulating EMT. So what I show you here is that NR2F2 is a key regulator of this malignant tumor state in SCC, and it do so by inhibiting differentiation, by sustaining the proliferative potential of these tumor cells, by promoting EMT and tumor progression. I didn't show you that, but it also decreases apoptosis and the recruitment of immune cells. So upon NR2F2 deletion, there's a strong infiltration of the tumor by macrophage and neutrophils, and it's also promoting angiogenesis and ECN remodeling. And you know, we really hope that targeting NR2F2 could make a difference in those kind of tumors. With that, I'll thank you for your attention, and I'll thank all the people who did this work. The first part of the work was done by Dani Nassar and Mathilde Latilde. The second part on the different EMT tumor states was done by Eugenia Pastochenko, as well as the FAT1 stories. And the last part was done by Federico Mori. And with that, again, I thank you for your attention, and I will be very happy to answer your question.
Thank you so much. That was really a master class about EMT and cancer biology. So please type your questions in the Q&A. I will start reading some that have already shown up here. So Thomas Tan is asking, do we know if the early or late EMT is more susceptible to TGF-beta pathway modulation?
That's an excellent question. Actually, we did not test that in the sense that in vitro, it's in our model, at least, it's not easy to stabilize the hybrid population. They had the tendency to progress to the very late EMT states. But, you know, it's not impossible that actually the modulation of the TGF might be more important in the late stage. Why I'm thinking that is when we check for the motif discovery in the ataxic peak, the SMAD2/3 motif were preferentially upregulated in the late EMT rather than the early EMT state. So that's the only piece of data that I can remember that would suggest that it's more, you know, a late EMT, but, you know, we should certainly test that directly.
Awesome. Ganendra Sain is asking, does SOX2 regulate YAP? Can they regulate each other?
We didn't see that as far as I remember, at least not transcriptionally. I'm not sure that we have done in SOX2 knockout the YAP staining, but I would expect that since YAP was already present in the absence of SOX2, that it would still be present in the nucleus there. And it certainly do not inhibit YAP translocation, that's for sure.
Yunhao Zai, thanks for a great talk. Does the EMT transition marker, so VCAM, CD51, CD61, can they be also used to define human EMT states? So we didn't try in the human model, in the human PDX. We found that to be, they also overexpress in the mammary tumors from P3CA and P53 deletion, and also in the MMTV PMYT, the EMT tumor cells also express this marker, but we haven't tried that on the human settings. Srinivas Vinod Saladi, what is the role or potential contribution of P63 in the FAT1 mutation context? P63 and YAP1 have shown to physically interact, so do you expect an enhanced transcription mediated by P63 and YAP1?
Definitively, yes. And so very, very very often actually SOX2 and P63 works together. They're in the same amplicon that can be amplifying certain type of non-small cell lung carcinoma. And as I show you, P63 was very strongly sustained in this human mutated for FAT1, and the motif of P63 was also found to be co-expressed with SOX2. So clearly, yes. And in previous work, we found that sustaining the expression of P63 can promote the epithelial differentiation or restrict TMT in those tumor cells. So I would say certainly yes.
Okay, awesome. I do have a few questions out there, more questions here. So follow up, any epigenetic factor contribution in the FAT1 mutation context apart from EZH2 and SOX2?
There were several other EMT regulators that were differentially expressed, but we didn't assess the function only directly for EZH2, so we didn't really investigate the function of the other one. But certainly, we have some data suggesting that other EMT regulators, such as, for example, NSD1, can also potentially promote EMT. And I've heard from my colleague that many epigenetic regulators seems to really control the different EMT trajectory as well. Yes.
So I do have a few questions. There's another one. Okay, read this one and then I ask mine. Do you have any suggestions for in vitro models like organoid or organoid on chip models to detect EMT?
So organoid model, are great. They also it's difficult to passage. We found that they are not also a little bit like upon transplantation. They cannot be always as stable as we would love to, and so such kind of states we haven't directly test this in human organoid with the most. It didn't really. It was not really helpful as compared to the 2D straight culture of the. But that maybe squamous epithelial grow very well in 2D. So maybe I have a bias for the 2D culture here, and possibly for other glandular epithelia, the 3D in organoid might be much better than for the squamous.
So now actually this in vitro question made me think of something. So it was so beautiful, the image that you showed, area 1, area 2, area 3, area 4, and you showed these different stages, how well how well they are separated in the tumor. And that suggests that there's a niche. I mean, you did mention something about the niche. So my question is, do you know what that is? And also thinking of your area 4, where you see more lymphatics and vessels and more immune cells, is that a consequence of that stage, or are they helping the tumor cells becoming that stage?
That's a great question. So clearly, we found that actually when you compare the late EMT, they overexpress tons of chemokine and cytokine that were predicted to recruit this inflammatory cells, in particular the macrophage. So in this model, we have treated the mice with anti-CFSR1A and anti-CCL2 during the time of tumor initiation. And we could clearly show that preventing or decreasing macrophage infiltration in this tumor switched the cells toward more epithelial characteristic and less EMT and more early EMT compared to the late EMT. Yes, clearly.
Wow. So it's a feedback.
Exactly. So they interact. So the late EMT recruit the macrophage, and the macrophage help them to sustain their EMT states, clearly.
That's beautiful. Have you checked, for example, MHC class 1 or antigen presentation in these cells? Do they show differences in their ability to kind of be seen by the immune system?
Yes, so that's something, but it's very the early time, but we do see at the single cell level, some population with different level of MHC class 1 and even class 2 in some of the cells. So yeah, there's clearly some, we haven't, dig that much for the moment in that topic, but we do see that by single cell RNA sequencing.
You have amazing, super elegant models, and I think, I'm biased, but there's so much immunology you could do in those tumors. I think it's beautiful.
So, but for that, I would be very happy to collaborate with you because, immunology is not my specialty, and we really need to have competence.
Yeah, no, of course. We can talk about. Oh, it's already 1:05. So if anyone has more questions, please type them. And we will make them available to Dr. Baupin. Thank you so much.
Thank you very much.
Yes, this was an amazing presentation. Thank you so much.
Thank you very much, and thank you very much all for attending. Bye. See you soon.
Thank you. Bye.
Bye.